Hello and welcome to this video series on ImageQuant TL Analysis software. In this video, we will guide you and help you get the most out of your images.
Welcome to this tutorial video of ImageQuant. In this video, we will look at the Gel and Blot Analysis module, and in particular the Purity function. First, click on the Gel and Blot Analysis button. In this video, we will look at the tutorial image ÄKTA. You find it under Open Tutorial Image, and there are a number of different tutorial videos that you can use to learn more about the software. In the first view, you see that this is a grayscale image which has the unit optical density. It was captured using the ImageQuant 800 imager, and you find all the capture settings in image information. Many of the mandatory steps have already been performed: the creation of lanes, background subtraction, band detection, and we have used custom settings in this case. But there are a number of presets: low intensity, high intensity, and default settings. So please find the optimal settings for your image. So these are all mandatory steps, and then there are a number of possible analysis steps: molecular weight, compare, normalize, and quantity calibration. But in this video we will focus on an analysis called Purity. And the purpose of this analysis is to present the data from a chromatogram together with a gel analysis. And the parameters that are of special interest is the band percent in the lane. It can also be expressed as the lane percent. And the difference is that the percentage is calculated based on all the bands in the lane or on all the lane. So also the non-band areas can be used in that calculation. So those percentage numbers are usually lower compared to the band percentage. One step here that you need to do manually is to import the chromatogram data from the ÄKTA run. This is an example of a purification of a HisTrap protein using a HisTrap column on an ÄKTA Pure chromatography system. And the data has already been imported. You can choose the number of channels that you would like to display in the figure. Let's dive deeper into the analysis. In this case, we can start by looking at the start material. It is clear that this is a very broad lane profile. It has a number of non-resolved protein bands of approximately equal intensity. There is no one single protein that stands out. But in the elution fraction, there is a clear peak, and that we can assume is the target protein. And if we then want to know what is the band percentage, we can see that this is band 11 in this lane, and the band percentage is 37.8. If we compare that to the start material, we see that the same band has a percentage of roughly 8.4. So there has been a significant purification using the HisTrap column on an ÄKTA Pure chromatography system. It is interesting to note that the band percentage is affected if the sample is diluted. So in this case, you see that the last lane is the diluted sample of the elution fraction 5 times. And in that case, the target protein appears to be more pure. It has a number of 57%. And that is actually showing the limitation of Coomassie staining. So if there is not enough sample to stain, that means that the weak proteins, the impurities, will fall below the detection limit. So if the user is not careful, it might appear that you have a purified protein, but you are actually not staining such low amounts of impurities if you dilute the sample. So please be a bit careful with dilution when you do Coomassie staining, and it is of course better to have more sample in your lane and to detect also the impurities well. Finally, we can have a look at the chromatogram view. It is easy to zoom in and out in this view, so say that you are interested in a particular peak. Then you can zoom in and out like this. You can move it just by clicking on Control and then right-clicking on the mouse. To zoom out, we just go to Reset Zoom, and you can include as many different parameters as you wish that you have imported in as part of the chromatogram. So normally when you are interested in the detection of your protein, then you look at the UV detectors. In this case, the 280 and the 310 nanometers. This concludes this video. Hopefully you will find it a useful feature to import chromatogram data to then have in the same report to calculate the band percentage values, and it is very easy to export the results as a PDF file. You generate the PDF report in the final step, or you can directly copy to clipboard or save to file from within the view that you are looking at. As a final note, for the import of chromatogram data, there are two file types that can be used. One is the .asc files. They need to be exported from the Unicorn Classic module. The other file type is .csv. They can be directly exported from the Unicorn Evaluation module, but please note that they need to be resaved with the right encoding. So there is a step-by-step guide to how to do this. Please request such a guide if it is not clear. You just open that file in Notepad and you then remove the CSV and resave with the UTF-8 encoding. Then it can be opened just like the ASC file. Also note that when you export chromatogram data from Unicorn, the x-axis 0 setting is important, especially if you want to display fractions correctly.
Thank you for watching this video. Make sure to check out the other videos in this series or go to cytiva.com/IQTL to learn more. See you in the next video.